So this is video 7, I think, of practical term sheet 1, and we're still in week 1. We're going to be covering the cells, the cell parts, and also the cell cycle. You'll notice if you look at your list that not only do you need to be able to identify them on a model, you have to identify the parts and stages and know functions. This means for every one of these organelles that are listed, you got to know what they do. Okay? So I'm going to be covering those kind of briefly. And when I say cell models, there's 3 different ones that I could be bringing out. So here they are, just to briefly introduce you. We got this guy here. We have 4 of these ones in particular. We also have this gray cell model. We only have 1 of him, but he's useful for different things. And we also have this one. So this one's actually part of this mitosis guy here, but he still has some cell parts that you can see. And all of these models show something a little different, and some of them have things that the other ones don't have. So know all 3 of these, because some of these have some stuff better than others, okay? So you'll see what I mean when I start going through. But just be aware that all 3 of these need to be on your docket. You got to know all 3 of them. Even if for your homework you're only labeling 1 of them, okay? All right, let's get started. First we have the nucleus, and you can see that on all 3 of these our nucleus looks pretty similar. We got this purple area here with the ball inside, purple area here with the ball inside, and we have this area here also with 2 balls inside. But the nucleus is basically where just all your genetic, and you've heard this a million times, but all your genetic information is stored, and it's kept segregated from the rest of the cell, okay? Making up the nucleus, just so I go in order here, we have the nuclear envelope. It's considered to be an envelope rather than a membrane because it's actually a double-layered membrane. And you can see that really nicely here. So here's again the nucleus, and you can actually see that there's a space or a hollow, and there's an outer membrane and an inner membrane. That's why it's called the nuclear envelope rather than the nuclear membrane. And inside, we have the nucleoplasm. So whenever you hear the word plasm, it usually means stuff. So it means like fluid and the genetic material that you're going to find in here. That's all going to be nucleoplasm, and it's associated proteins that you'll find in there as well. Chromatin is going to be the loose DNA with its associated proteins. So you can see in here that there's like this coppery frizzy stuff. That's going to be representing your chromatin. Okay, that's your chromatin. Unfortunately, you can't really see those in these other models very well, but you can see that it's kind of loosey-goosey in here as opposed to, for example, this guy where we have fully condensed genetic material. This is all loosey-goosey. It's chromatin, DNA with its associated protein. Okay, and in this one, you can't really see anything going on in there, but just know that it's in there, just so I don't miss anything. If you have your chromatin and it condenses, you get chromosomes. There you go. And you have to just know the numbers. So you have to know that there's 46 chromosomes or 23 pairs. Okay? So that's all we really want you to know for that.
In humans.
In humans. Plants can have like a ton more or even fewer sometimes. And I think Drosophila only has 4, right? Is that 4 just separate ones or 4 pairs? I forget. Anyway, different critters have different numbers. Different plants have different numbers. But for humans in particular, we have 23 pairs or 46 total. Okay? Cool. Moving on, we have the nucleolus, and on your list it says nucleoli, so that just is plural. So nucleolus, singular, nucleoli, it's plural. So the nucleolus, in this case, is going to be this round structure here, okay? And what is the job of the nucleolus? This is where ribosomes are manufactured, okay? So there you go. There's your nucleolus, and that particular one. This one, for some reason, has 2 nucleoli. I don't know if that's like a legit thing that occurs, but there you go. So again, what's the job of the nucleolus? It is the factory for where ribosomes are made. Okay? So ribosomes can be made, and then they can be transported through these holes that are in the nuclear envelope. And then life is good. Moving on. We have the cytosol and the cytoplasm. So the cytosol and the cytoplasm are different from each other. The cytoplasm is going to be the fluid, the proteins, all the stuff, okay, outside of the nucleus, obviously. But the cytoplasm is going to be all the fluid and all the stuff. Cytosol is fluid only, okay? So if I were to ask you on a quiz or a practical, or if Anna were to ask you on a quiz or practical about cytosol, we'd put like a sticker here, for example, okay? So that'd be cytosol. Cytoplasm might be more like a question format, like, you know, what is all the fluid and stuff inside of a cell called? So that's going to be the cytoplasm, okay? And again, same ordeal, cytosol versus cytoplasm, okay? And same deal here. Cytosol, where there's like just a blank spot or cytoplasm. Moving on here. Ribosomes. So I already said that the nucleolus makes ribosomes. What are those and where can you see them? So a ribosome is very small organelle. It's like a little speck in a cell. So sometimes when you see cell models, you'll see that they look kind of speckled, and that's usually indicating ribosomes. In this case, ribosomes are going to be these little red sprinkles. Okay, so those are ribosomes. Ribosome's job in life makes proteins. Okay, so it takes messenger RNA, translates that baby up into a string of amino acids, which will be a protein. So that's our ribosomes' jobs here. Okay. On this particular model, ribosomes are indicated by these little tiny pock marks. So you can kind of see ribosomes in there, all right? Now on this other model, however, you can't see ribosomes at all. So again, there's 3 models you gotta know. They all have something different, so just keep in mind. So there's ribosomes. Their job is to make proteins. Next we have the endoplasmic reticulum. The endoplasmic reticulum usually is depicted as flattened sacs in the cell. So again, kind of like with our nuclear envelope here, we have 2 membranes. Well, it's a continuous membrane that wraps around, but it's kind of like sandwiched so that there's space on the inside. Okay, so it's an enclosed sac. The endoplasmic reticulum, in my mind, its most important job is storing calcium. That's not all it does. It also does help with lipid synthesis, and it can help with modification of proteins. You'll notice that some of this endoplasmic reticulum has ribosomes stuck to it. This is considered to be rough endoplasmic reticulum, or RER. We do expect you to spell it out entirely on the practical, so no being lazy and putting RER. We won't accept it, okay? So anyway, ribosomes are stuck to the surface here. They can actually deposit proteins directly into the endoplasmic reticulum, and they can be modified within those flattened sacs. On these other models, however, if it doesn't have ribosomes, it's going to be smooth endoplasmic reticulum. And you can actually see on here that there is smooth. So rough has ribosomes, smooth has nothing on it. On these other models, they're very difficult to tell apart. You can see that anywhere where you see endoplasmic reticulum, in the case of this particular model, kind of see where it's like ribbony looking, almost like bacon or something. I don't know, it's a terrible example. Anyway, these here where it's still the same color, like this gray color, and it's like kind of ribbony, that's going to be your endoplasmic reticulum. And if you look at all of these in this particular model, they all kind of have those pocked marks like ribosomes. So there's no good way to dissociate smooth from rough endoplasmic reticulum on this model. We would not use this model to ask you for smooth or rough endoplasmic reticulum. We would only use that other model where you can see clearly the ribosomes and the difference between rough and smooth. Okay? On this particular model here, you can't see ribosomes at all, like I said. So you can't distinguish rough from smooth once again. All right? So just keep in mind, like I said, certain models are better for certain things. Okie dokie. Moving on, we have the Golgi apparatus. So they're going to look hugely different from model to model, so definitely pay attention here. On this particular model, Golgi apparatus is this red squiggly guy. Okay, he's kind of similar to the endoplasmic reticulum in that it's a series of, it's like an enclosed sac. It's like kind of looks pancakey almost if you cut it in half. So again, it has like a space in the middle and it's bound by membrane all the way around. So it's kind of similar in that regard, but it does different stuff. In this case, it actually helps to ship stuff around the cell. So you've probably heard it before, the FedEx of the cell or whatever, right? On this particular model, this is going to be your Golgi apparatus. This is going to be your Golgi apparatus. So this is like a 3-dimensional view. This is if you cut it in half. So remember I was saying like if you cut it in half, it kind of looks like endoplasmic reticulum because it's those flattened sacs. Oops, there you go. And usually around the Golgi apparatus, you're going to see indications of little vesicles coming off of it. That's because, like I said, it's kind of that FedEx of the cell, and the way it's sending stuff around is in these little vesicles here, okay? And you can even see up here that there's some stuff being exocytosed. So sometimes that comes off of the Golgi apparatus. A vesicle will come out here, join up with the plasma membrane, which is the outside membrane here of the cell, which we'll talk about, and exocytose stuff. In this particular model, the Golgi apparatus is going to be these red pancakey guys here. And again, you can see the indication of vesicles coming off of here. So again, it's actively transporting stuff around the cell. Okay, that's your Golgi apparatus. Moving on, we've got lysosomes. In this case, there's only one model that's going to show lysosomes clearly. That's this guy. So lysosomes are kind of like the stomach for the cell. They're going to digest stuff. In this case, usually stuff it doesn't want anymore. So in that case, it's not like the stomach because we want to digest stuff that we want to eat and break down for later. So lysosomes on this particular model are going to be these gray guys here, okay? And you can actually see he's got like a little, I don't know what that is, but it's a thing, right? So you got a thing on there versus like these smooth guys. So yeah, this here is going to be a lysosome. These actually have proton pumps on the outside, so they pump hydrogen into this vesicle here, the lysosome, and that decreases the pH or increases the acidity, however you want to think about that, and it can break down proteins and even bacteria. So they're like little digesting guys here, just like the stomach has acid in it. Anyway, there you go, lysosome. But you can only really see it on this particular model, the gray one. We also have, I don't want to go out of order. Good. We also have the peroxisomes. You can only see peroxisomes on this model. It's right here, this little yellow guy. So peroxisomes are going to deal with the byproducts of lipid breakdown or fatty acid breakdown. When you break down fatty acids, you get H2O2 or hydrogen peroxide. Hydrogen peroxide is considered a free radical, so it can affect proteins and other things inside of the cell. It's no bueno. You don't want that. So you got the peroxisomes here. They take that H2O2 and they break it down into harmless oxygen and water. Life is good, right? So there you go, peroxisomes. That's its job and what it looks like. And again, you will only see it on this particular model. Moving on, we got mitochondria. So mitochondria you will see on every cell model here, but it looks different, kind of like how the ER looked different on all of these, okay? So mitochondria in general, you've probably seen on pictures and illustrations that it kind of looks like a jelly bean or a kidney bean. So here on this particular model, kind of looks like these orange Tic Tacs here, but you can see that some of them are more bean-shaped as well. One great way to tell if you're looking at mitochondria or not is they'll almost always give you a half view. And in this half view, it's got this squiggly inside. So you can see that this has like this squiggly shape on the inside. These are referred to as cisternae. You don't need to know that, it's not on your list, but just so you know. But it's got these folds or twisties. So this is the outside of the mitochondria. So it's round and smooth on the outside, but if you cut it in half, this is what the insides look like, okay? On this particular model. On the gray model, our mitochondria look like kidney beans. So again, here you can see the smooth surface, round, three-dimensional. If you take that mitochondria and you cut it in half, it's got the squigglies on the inside, just like I was mentioning before, those cisternae, right? Lastly, for mitochondria, this is the weird one. I always think that this mitochondria kind of looks like worms or something, but this is legit a mitochondria, okay? So you can see that it's this round, three-dimensional kind of shape. Rather than showing you a half of a mitochondria, they just cut away into it. So you can see, again, that twisty-turny shape, the cisternae, right? So what's the job of the mitochondria? You've heard this a million times. The powerhouse, the powerhouse of the cell. The mitochondria produces ATP. So ATP helps the cell do all kinds of jobs. In the case of muscle, for example, which you guys will learn about later, ATP helps with contraction. So your muscle does not move unless you have mitochondria producing ATP. So it's pretty important. Moving on, we got centrioles. Centrioles, to me, always look like cut-up pieces of licorice or like cut-up Twizzlers. The job of the centriole is to help pretty much almost, I'm not going to say exclusively, but almost pretty much all it does is help with mitosis. I don't know if Anna has a different opinion on that, but anyway, maybe I'm not the right person to ask about that. But usually the centrioles will come in like the way that they're depicted is like a standing up cut piece of Twizzler and then one that's laying down, okay? So this whole thing here is considered to be the centrioles. There's acentriole, there you go. Okay, and what they do is they actually extend out filaments, in this case microtubules, and when the cell is going to try and divide, there we go, it actually duplicates those centrioles. So here, like I said, are the centrioles, these yellow cut up pieces of Twizzlers. You can see that it's actually duplicated when you're going to do mitosis, and you can see indicated on here that it's extending out fibers, or in this case microtubules, and it's actually going to snatch on to these chromosomes, okay? So just so that you know what they're doing and kind of what they look like. But when the cell's not trying to divide, it's just these guys right here, and they extend out some of microtubules. On the other models, again, they just kind of look like these cut up Twizzlers. So right here, there's your centrioles, right?
Tasty.
One that's laying over, one that's standing up. Lastly, for centrioles, it's weird on this model, but rather than being a three-dimensional cut-up Twizzler, it's this thing here where you see there's these lines, and then as if the other one were laying down, you're looking into it, okay? So there's your centrioles in that particular one. I don't like how this model looks for the centrioles, so usually I'll use this guy or one of these guys so you can see the centrioles more clearly. Just keep in mind that whenever we do make a quiz or a practical, we're picking the model we think shows that thing the best, right? So we're not just throwing anything out and be like, I don't know, this one kind of sucks, but like, I don't know what it is anyway. We're always going to pick what we think looks the best, okay? All right, moving on, cytoskeleton. Now, cytoskeleton you can't really see on these guys. You just have to know that it's just a network of proteins that's going to be helping the cell maintain its shape. Best example of this is for erythrocytes or red blood cells. You guys should know that they kind of look like little inner tubes. It has a very rigid cytoskeleton that helps the red blood cell keep its shape. So, cytoskeleton, that'll be more a question format, like what's the proteinous network that helps the cell maintain its shape? Cytoskeleton, okay? Moving on, we have the plasma membrane. Now I know I mentioned the plasma membrane a few times and I didn't really cover it as its own term. The plasma membrane is just a, you've heard this before too, a lipid bilayer, and it encapsulates the entire cell. So here we go, here's our cell, and all along the outside here there's actually a fatty layer that is kind of containing everything inside. Usually it has, it's studded with proteins, but you don't really need to know about that for the scope of this course. But the plasma membrane, if I want to identify it to you, I will put a sticker on the outside. Okay, so whether it's this or this, okay, so on the outside of that, that's the plasma membrane. And the question will be something along the lines of what structure is indicated by this sticker. Moving on. Next we have cilia and flagella on the list. We do not have models of these guys, so again, that would be a question format. So cilia is going to be an actual extension of the cell itself. So your plasma membrane will actually be kind of forced out. Here we go. I can draw it real quick. So if you are a cell, you have your plasma membrane like this. If you have a cilia, the cilia is actually an extension, kind of, like that. So it's actually filled with cytoplasm and cytosol. And these can be motile, and they can whip around and help move cell, which is great. A flagella, on the other hand, is not an extension of the cell itself. It's, in other words, not an extension of its membrane. It actually has like a little motor, and it's just a protein chain off of the end. So you can tell right away that the only flagellated cell in the human body can be human sperm. Okay, so only males have that, which, there you go. But anyway, that's flagella versus cilia. So cilia you can find wherever, so you need to move stuff along. So for example, in the trachea, you need to move mucus along, so those cells are often ciliated. Or in the nose, you need to move mucus along, so those cells are often ciliated as well. And that's it for cell parts.